students, welcome to ECNUS where nursing education made easy. Exams are coming, right? Are you worried about your exams? Don't worry. We ECNUS team is here to help you. So we are ECNUS here to help you to ease your exam tension. So don't worry, listen to the class and get an update on your knowledge and write the exam in confidence. So let's start. Today we are going to discuss about some important topics and questions from applied microbiology and infection control and safety. So let's discuss one by one what all the important topics that are coming from this subject. So let's start. Yes. So first we are going to discuss about what is microbiology. So let's start with the definition itself. What is microbiology? So what is microbiology? The study of microorganisms and their role in human health. So it's nothing but the study of microorganisms and their role in human health and disease. So that's the definition of microbiology, which are visible under microscope. So with our naked eyes, we can't able to see some kind of microorganisms that is producing the disease. So microbiology is the study of microorganisms which are not able to see with their naked eyes and it is only visible through the microscope. So this is the definition of microbiology. I hope you are getting the point. So let's continue. Microbiology includes different studies. So what are the studies microbiology is combining? It includes bacteriology, the study of bacteria, virology, the study of viruses, and parasitology, the study about parasites, and also mycology, the study of fungi. So this is the microbiology is divided into these subdivisions. So it is bacteriology, virology, parasitology, and mycology. So what is the definition? Once again, we will see the definition for you just to remember. So what it is? Microbiology is the study of microorganisms and their role in human health and disease, which is only visible under microscope. So let's move on to the next question and next topic. So now we will see what is the importance and relevance of microbiology to nursing. So why as a nursing student we need to study microbiology. So what is the importance of microbiology? So let's discuss. To promote health. So as a nursing professional, we are giving the care to the patient. So to promote their health, we need to study about microbiology. To promote health, to diagnose and treatment infections, that is by bacteria, virus or any fungal infection. So we need to diagnose and treatment. Sterilization and infection control. So in the operation theatre or in the ward or in the procedure room, do the sterilization and infection control. We need to know about the principles of microbiology. Prophylaxis. It also uses a prophylaxis to prevent diseases. And diagnosis and control of hospital infections. Also to help understand importance of biomedical waste management. So these are the importance and relevance of microbiology to nursing. Clear? Let's move on to the next question. So here we will discuss about next important question, that is Robert Koch. What is this? Robert Koch. Who is Robert Koch and what is the importance of Robert Koch postulates? Let's deeply discuss. So who is Robert Koch? He is the father of bacteriology. He introduced the staining technique. So the staining technique to identify the bacteria. So he introduced that staining techniques. Also introduced solid media for culture of bacteria. To do the culture of bacteria in laboratory, he introduced the solid media. Hanging drop method. So he also introduced hanging drop method, discovered the Anthrax bacillus, tuberculous bacillus and Vibrio cholera. So he identified some microorganisms and discovered some microorganisms. So what are the microorganisms it is? Discovered Anthrax bacillus, tuberculous bacillus and Vibrio cholera. So now we will see Koch postulates. So it's very important. So it's a short answer type question. So you'll get for a 2 mark or maybe you will expect this as a 5 mark. So what is Koch postulates? Let's see. So Koch proved that microorganism can accepted as a causative agent of an infectious disease by following conditions should be fulfilled. So what are the conditions? First one is the microorganism should be constantly associated with the lesion of disease. So the microorganism should be constantly associated with that particular infection. So that's the number one postulate. So the microorganism should constantly associated with the lesions of that particular disease. Next one is It should be possible to isolate the organism in pure culture from lesion of disease. So we are taking out the lesion, that from the lesion we are taking a sample. So in the pure culture, if we are doing the pure culture, we can able to isolate the organism, then it is related to that disease. So it should be possible. It should be possible to isolate the organism. Isolate the organism in pure culture from the lesion of that particular disease. Next one is, the same disease must result when the isolated microorganism is inoculated into a suitable laboratory animal. So from that microorganism, from that lesion of that disease is inoculated into a microorganism, inoculated into an animal, laboratory animal, and the symptoms is producing, same symptoms is producing inside the animal also, then we can say that it is related to that disease condition. So the last one is, it should be possible to re-isolate the organism. Again, we can able to re-isolate that organism in pure culture from the lesion produced in the experimental animals. So these are in the experimental animals, again that symptom should be present and we can able to re-isolate that microorganism. Then this is known as, that microorganism is related to that particular disease. So this is Koch postulates. So what are the Koch postulates? The microorganism should be constantly associated with the lesions of disease. It should be possible to isolate the organism in pure culture from the lesion of disease. The same disease must result when the isolated microorganism is inoculated into a suitable laboratory animal. It should be possible to re-isolate the organism in pure culture from the lesion produced in experimental animals. So this is Koch postulates. Clear? Let's move on to the next question. So next we are discussing about principles of microbiology. So what are the main principles of microbiology? Let's see. So first one is cell theory. All living things are made up of cells. So it is mainly based on the cell theory that all the living things are made up of cells. Next is microorganisms are everywhere. So it is found in air, water, soil and in living beings. Classification of microbes. It is mainly classified into bacteria, virus, fungi, protozoa and algae. So growth and reproduction. Microbes grow and reproduce rapidly. So the growth and reproduction will be rapidly, and that is microbes will grow and reproduce rapidly. So very fastly they reproduce. Metabolism. Microbes carry out chemical reactions to survive. So they will have a chemical reactions for their survival. Genetics. Microbes pass through the trait of DNA or RNA. So it's involved in genetics also. Pathogenicity. Some microbes causes diseases. Immunity. The body fights microorganisms through our immune system. So if one microorganism is entering our body, our immune system start to fight against that. So the symptoms will be there. So fever and all we will get. So this is immunity. The body fights microorganisms through the immune system. Sterilization and disinfection. That is the method to kill or remove microbes. Usefulness of microbes. Used in medicine, food, industry, like antibiotics and yogurt. So microbes is used in medicine, food and industry. So this is the principles of microbiology. So now let's discuss about parts of microscope. So what are the main parts of microscope? So we will see first there is a eyepiece. It is ocular lens will be there. One arm to hold that where will be one arm. Nose piece will be there. Objective lenses. So through these lenses only we can able to see the microorganism. So stage will be there. Condenser and diaphragm. Light source will be there. And here there will be a coarse focus and fine focus will be there to adjust the size. We can able to see the bacteria or microorganism through adjusting the size, and there is a base. So these are the main parts of microscope. Now we will discuss about ultrastructure of bacterial cells. So another important. So there is a chance of question they will come, draw the bacterial structure, bacterial cell wall structure. So there will be in bacterial cell wall structure, there is cytoplasms will be present, ribosomes, nucleoid, DNA and flagella. So these are the flagellas. Attachment, pili will be there, plasmid will be there, sex pulus will be there, cell membrane, cell wall and capsule. So these are the main structure of a bacterial cell. So you need to draw in your exam. So draw neatly and mark this part. So it will be very helpful you to score the 5 or either the 2 marks. So this is another example, like capsule will be there, flagella, cell membrane, chromosome will be present, meiosomes will be there, protoplasm, villi will be there, cell wall and pyloribosome. So these are the main structure of a bacterial cell. Next, we are moving to the next important question, bacterial growth curve. So what is bacterial growth curve? What are the main growth curve? What are the main phases in the bacterial growth curve? So this is the number of cells that is coming in the log, number of cells in log and the time. So there will be lag phase. So there is a lag phase will be there, and there is exponential phase that is also known as log phase. So you need to draw this in your exam answer sheet. So this is, if your question is draw the neat diagram of bacterial growth curve, then you need to do this. Number of cells, lag phase, log phase, stationary phase. So there will be stationary phase. So this is the stationary phase, and a decline in the phase, that is death phase. So this is the 4 phases of a bacterial growth curve. It is lag phase, log phase, stationary phase and death or decline phase. Clear about the points? So now let's move on to the classification of microbes. So that is the next question, classification of microbes. So how it is classified into? First one is bacteria. It's a single cell prokaryotic. So bacteria is a single cell prokaryotic. Its shapes are Koch. So Koch means it will be in round shape. Bacillus means rod shape. Spirilla means in spiral shape. So this is the bacteria. It's a single celled prokaryotic. Now coming to the viruses, it is a non-living outside host. So the host will be, it is non-living outside host and need host cells to reproduce. So it should be, it's to produce, they need to reproduce, they need the host cell. Example, HIV and influenza, human immunodeficiency virus and influenza virus. Fungi can be unicellular or multicellular. It's either unicellular like yeast or multicellular like mold. Next is eukaryotic, causes infections like ringworm and candidiasis. So what are the fungi infections? Ringworm and candidiasis. Protozoa, it is another category is protozoa, that is unicellular and eukaryotic. So it will be found in the water and soil. Examples, amoeba and plasmodium. So you are familiar with the examples like amoeba and plasmodium. Next category is algae. So what is here? Most aquatic and photosynthetic. It will be mostly in aquatic nature and photosynthetic. Can be unicellular or multicellular. Not usually harmful, but sometimes causes algal blooms. So it sometimes causes or rarely algal blooms. Next category is helminthics or parasitic worms. It's a multicellular organism and examples are roundworm or flatworm. So this is the classification: bacteria, virus, fungi, protozoa, algae and helminths. Next we will discuss about size of bacteria. So bacteria are very small, measured in micrometers. So what is bacteria? It is very small and measured in micrometers. Its size range from 0.2 to 10 micrometer. So its size will be 0.2 to 10 micrometer. So example, E. coli. E. coli will be 1 to 2 will be the micrometer. Mycoplasma is 0.2 micrometer, the smallest. So this is the bacteria size. Size of bacteria is very small, measured in micrometers, range from 0.2 to 10 micrometer. Next we will discuss forms or shapes of bacteria. So what are the main shape? Kochi or spherical shape. Kochi spherical. Example, Staphylococcus bacteria and Streptococcus. Staphylococcus and Streptococcus will be in Kochi. Bacilli or rod shaped. Example, E. coli or Bacillus anthracis. Spirilla or spiral shape. Example, Spirillum. Vibrio or comma shaped. It will be comma shape. Example, Vibrio cholerae. Spirochetes, flexible spiral. Example, Treponema pallidum. So these are the forms or shapes of bacteria with examples. So next important question that may come for us a long essay or a short essay or either a short answer, that is gram staining. So what is gram staining? It's a method to use to classify bacteria in two groups based on their cell wall structure. So it is the method to classify bacteria based on their cell wall structure. So that is gram staining. So it is mainly classified into gram-positive bacteria or gram-negative bacteria. So what is gram-positive? So it will be color after staining. So after the staining process, their color will be purple or violet. What is the cell wall structure? Thick polypeptidoglycan layer will be there. So here in the cell wall structure, in case of gram-positive, there will be thick peptidoglycan layer. Coming to examples, it is Staphylococcus aureus. So the example for gram-positive bacteria is Staphylococcus aureus. So coming to the next category, that is gram-negative bacteria. So in the gram-negative here, after the color staining, it will be pink or reddish in color. So what are the features will be there? Thin peptidoglycan plus outer membrane. So there is an outer membrane. Also the peptidoglycan will be thin in compared to gram-positive bacteria. So examples are E. coli bacteria or Salmonella bacteria, Salmonella typhi. So these are the examples for gram staining. So what is gram staining? It's a method to classify bacteria in two groups based on their cell wall structure. Clear? So next move on to next question, that is culture media. So what is culture media? It is a culture media is a nutrient rich substance used to grow microorganism in lab. So it's where the microorganism is used. We are using this here nutrient rich substance to grow the microorganism in laboratory for study or diagnosis purpose. So this is culture media. So what is the culture media definition? Let's see again. So it's a culture media is a nutrient rich substance used to grow microorganism in the lab. So we'll see about it is divided into based on the physical state. There are solid, liquid and semi-solid. So based on the physical state. So what is solid? It contains agar. It's a firm surface and it is example is nutrient agar. Next is liquid classification. So liquid type no agar used on broth cultures and it is nutrient broth. Example is nutrient broth. Next is semi-solid category. Low agar used for motility test and it is motility agar. So this is based on the physical state. The culture media is defined into three types: solid, liquid and semi-solid. Now based on the function, culture media is defined into simple media, enriched, selective, differential, transport and anaerobic. Let's see one by one. So based on the function, first one is simple media, grows non-fastidious bacteria. So why we are using, what is the purpose of using simple media? To grow non-fastidious bacteria. Example, nutrient agar or broth. So next category is enriched media, extra nutrients for fastidious organisms. So it is used in the case of purpose of using enriched media is to extra nutrients for fastidious organism. It is examples are blood agar or chocolate agar. Next is selective media, allow only specific microbes to grow. So selective means selectively only specific microorganism will grow in this culture media. Example, McConney's agar or it is gram negative. Next is differential media, shows difference between organisms. So in this culture media we can able to see the difference between the microorganisms and like example McConney agar, lactose fermenters pink. Next is transport media, keep microbes alive during transport. So it's used in the keeping microbes in while transporting alive during the transport. Like stewards or amines media or steward media is an example for a transport media. Next is anaerobic media, grows bacteria without oxygen. So without oxygen we can grow the bacteria in anaerobic media. Example, Robertson's cooked meat broth. So this is based on the function. The culture media is defined into mainly simple media, enriched media, selective media, differential media, transport media and also anaerobic media. Clear the points? Yes. So let's move on to next one, that is live vaccines. Also it is known as attenuated vaccine. So you will get this question for a 2 marker or for a MCQ question. So what is live vaccines? It contain weakened microorganisms. So it will containing the weakened microbes or microbes will be present, give strong long lasting immunity to our body. So live attenuated vaccines will contain weakened microbes. So it will give strong long lasting immunity not for weak immune system. So it's mainly not for the weak immune system. So what are the examples of live attenuated vaccine? So what are the examples? MMR, mumps, measles and rubella, BCG, Bacillus calmette-guerin, OPV, oral polio vaccine, varicella zoster vaccine for the yellow fever vaccines and rotavirus vaccine. So these are the examples of live vaccine, or also known as attenuated vaccines. Clear? Yes. Now let's move on to killed vaccines or inactivated vaccines. So what are the examples? It is containing dead microbes. So in the killed vaccines, what will be there? Dead microbes will be present. Safe for weak immune systems, may need booster doses. So this may need booster doses. Example, IPV, inactivated polio vaccine, hepatitis A vaccine, rabies vaccine, influenza or typhoid vaccine. So these are the examples for killed vaccines or inactivated vaccines. So next important topic, that is cold chain. So what is cold chain? It's a temperature controlled supply system. It's a temperature controlled supply system to store, transport vaccine. So it is mainly used to do what? To store and transport vaccines from the manufacture point to the use point. From the manufacture to the point of use. So from where it is producing, from there to where it is using, that till that point we need to transport and storage vaccines. For that we have temperature control supply system, that is known as cold chain. So it is temperature constant will be, that temperature will be, it will be 2 degree to 8 degree. So from the manufacture point to till the point of use we are using vaccines to store. Vaccines should be stored and transported. So for that we are using a temperature control supply system with the temperature of 2 degree Celsius to 8 degrees Celsius. That is what known as cold chain. Clear? Cold chain. So now we will see next important topic, that is, or the next important question, what is ELISA? So what is ELISA? It is stands for enzyme linked immunosorbent assay. So it is standing for, ELISA stands for enzyme linked immunosorbent assay. So used to diagnose infections, hormones and antibodies. So it's mainly used to diagnose some kind of infections, hormones and antibodies. Like example, HIV or AIDS, we can able to identify or diagnose through the using ELISA test. Based on antigen antibody reaction. So this test is using antigen antibody reaction. An enzyme is linked to an antibody, produce color change if target is present. So there will be color change if any enzyme is linked to an antibody, there will be color change. So ELISA is mainly used in the cases of, like example, HIV or AIDS. Next we will see about the structure of IgE and IgG. So what is the structure? You need to draw the structure. So the strands you need to draw in your exam. So exams, it's a common question to draw the structure of IgE or IgG. So this is the structure. So there are carbon dioxide chains will be there, CH4, CH3, CH2. Here it is the chain you need to draw, CH1. So this is a double helix. Here you can see this is the light chain and this is the heavy chain. So draw this and identify, that is also an important topic. So try to draw in your draft sheet, draw before going to the exam. So next is Ig structure. So this is also, there is a light chain, heavy chain. So this is the heavy chain and this is a light chain. This is variable region is present. So there is a constant region. So this is constant region, but this is variant region and antigen antibody site. So antigen binding site. So this is the IgG structure. Now we are going to discuss about branches of microbiology. That is also one will come for a 2 marker or a 5 marker. So what are the main branches of microbiology? Bacteriology, to study about bacteria. Virology, studying about viruses. Mycology, it's studying about fungi. So what is studying about fungi? It is mycology. Parasitology, studies about parasites. Immunology is the study of immune system. Protozoology is the study of protozoa. Phycology, or it's also known as algology, the study of algae. Microbial genetics, study of genes in microbes. Microbial physiology, study of microbial functions. Medical microbiology is the study of microbes in human disease. Industrial microbiology, uses of microbes into industry, microbes in industries. Environmental microbiology is study of microorganisms in environment. Food microbiology is the study of microbes in food like spoilage or fermentation. So these are the main branches of microbiology. Next question we will discuss about Streptococcus pyogenes or group A beta hemolytic Streptococcus. So let's discuss about Streptococcus pyogenes. It's virulence factor. So what is virulence factor here? This help the bacteria cause infection. So there is M protein. So inhibiting the phagocytosis. So in the case of Streptococcus pyogenes, virulence factor. So there are M proteins that inhibit the phagocytosis, helps in adhesion to host cells. Next is streptolysin O and S will be there. Streptolysin O. It is oxygen label, destroying the RBC and WBC. Streptolysin O. Streptolysin S. Oxygen stable, contributes to beta hemolysis. Next is erythrogenic or pyogenic toxins will be present, causes fever and rash like in scarlet fever. Hyaluronicide, so it is nothing but break down connective tissue and help in spreading of infection. Streptokinase that dissolve the clot and aid in bacterial spread inside the body. DNase that is break down the DNA in pus, helping the bacteria move. So this is the virulence factor of Streptococcus pyogenes. Next we will discuss laboratory diagnosis of Streptococcus pyogenes. How we can able to diagnose that through laboratory. So first is sample collection. So from where the sample will be collected? Throat swab, pus or blood. Then microscopy. Gram-positive cocaine chains is seen in stained smear. So through the microscopy we can able to see the gram-positive cocaine. Culture. Grown on blood agar. So where the culture we can do, it's grown on the blood agar. Shows beta hemolysis, clear zone around the colony. So the bacterial colony should be clear zone. Bactericin sensitivity test. So Streptococcus pyogenes is sensitive to bactericin test used for identification. Rapid antigen detection test. So we can use to quickly identify from the throat swab. Serology. Anti-streptolysin O titer raised in recent infections. Like in the cases of rheumatic heart disease, we can do ASO test, that is anti-streptolysin O titer test. So these are the laboratory diagnosis for Streptococcus pyogenes. Yes, next important procedure that may come for the exam is agglutination. So what is agglutination? It is the clumping of antigen with antibodies. So the clumping of antigens with antibodies, that is not what known as agglutination, the process. It occurs when antibody binds to antigen on the surface particles like cell or bacteria. So the antibody will bind to antigen at the surface of particles like cells or bacteria. So the agglutination is the clumping of antigens with antibodies. So what are the types here? So slide agglutination for quick test in slide. Example, Vidal test. Vidal test we will use in the case of typhoid. So slide agglutination. Then next is tube agglutination, done in tubes giving titers. Also passive agglutination, antigens on lattice beds. Example, RA test. Reverse passive, antibodies on beads to detect antigens. Hem agglutination. So here clumping of RBCs. Example, in the case of blood grouping. Koch agglutination uses Staphylococcus aureus with antibodies. So this is the examples or types of agglutination, the process. Here the clumping of antigens with antibodies. So next we are moving to the next question, that is bacterial spore. So this is the structure of bacterial spore. There is a core will be present, and after that inner membrane will be there, germ cell wall will be there, cortex will be there, outer membrane will be there, and a spore coat will be there, and exporium. So this is the structure of a bacterial spore. So there is a question that may come for the exam. Draw the structure of a bacterial spore. So this is the structure of bacterial spore. Core containing DNA and enzymes. So this core will containing the DNA and enzymes will be there. Cortex is the thick protective layer. So cortex will protect covering the bacterial spore. It's a thick protective layer. Spore coat. So what is spore coat? tough outer protein layer. So it's a protein layer that is tough outer protein, and exporium, it's outer thinner layer. So it is the outer thinner layer of a bacterial spore. So this is the structure of a bacterial spore. Yes, so I hope everyone is getting the point. So we are continuing to the next topic, bacterial modes of nutrition. So what is the bacterial mode of nutrition? It says autotrophic, so make their own food. So what is categorized into autotrophic? It is making their own food. So that category of bacteria is known as autotrophic, uses sunlight, photosynthetic, or chemical like chemosynthetics. Examples, cyanobacteria, that is photosynthetic in nature. So they'll use their food, they'll make for their nutrition, they'll make their own food. Next is heterotrophic, get food from organic sources. So the heterotrophic, what happens here? Getting food from the organic sources. Like types, saprophytic, that is feeding on dead matter. So saprophytic bacteria will feed on the dead matter. Parasitic, feed on living host. Symbiotic, it lives mutually within the organism. So this is the types, bacterial mode of nutrition, where the bacteria is getting the nutrition, autotrophic or heterotrophic in nature. So next we will discuss about BCG vaccine. So it was a 2-marker question for the last year. So what is BCG vaccine? Full name is, what is the BCG stands for? Bacillus calmette and guerin vaccine. BCG stands for Bacillus calmette and guerin. What is the purpose? To protect against tuberculosis. So BCG vaccine is used to protect against tuberculosis. It's a live attenuated vaccine. So it is from weakened Mycobacteria movis. So it is a category, it's live attenuated vaccine. Given to newborns and infants, especially in countries with high tuberculosis. So it's given to newborn babies and to infants. Next is route, usually given by intradermal injection. So it is an ID, intradermal injection under the skin. What is the effectiveness? Provides protection mainly against severe forms of tuberculosis in children like TB meningitis or miliary tuberculosis. So it is BCG. So what is BCG? Bacillus calmette and guerin. It's a live attenuated vaccine used against TB and given to the newborn babies and infants. Yes, so clear with BCG vaccine. So we'll continue with infection control and safety. The first question, that is hospital acquired infection. So what is hospital acquired infection? It is also known as nosocomial infection. So what is nosocomial infection? It's an infection that the patient acquires while receiving treatment in a hospital. So the patient is already admitted to the hospital with a disease condition, and the patient is developing another disease condition from the hospital, then it is known as nosocomial infection. So hospital acquired infection is also known as nosocomial infection. It's an infection that the patient acquires while receiving treatment in hospital or healthcare facility that was not present or incubating at the time of admission. So at the time of admission, that patient doesn't had that disease. It was after the admission to the hospital, after 48 hours, that person is having that another disease symptoms. Then it is nosocomial infection or hospital acquired infection. This infection typically appears 48 hours after or more after the hospital admission or within certain period after discharge. So at the time of coming to the hospital for the admission, that patient doesn't have that symptoms of particular disease. So after coming to the hospital, after getting admitted, after 48 hours, they are developing another disease conditions, other diseasing symptoms. Then it is known as hospital acquired infection. So let's see again one more time the definition. So hospital acquired infection, also known as nosocomial infection, is an infection that the patient acquires while receiving treatment in a hospital or healthcare facility that was not present or incubating at the time of admission. These infections typically appear 48 hours or more after the hospital admission or within a certain period after discharge. So this is the definition of hospital acquired infection, also known as nosocomial infection. So what are the common type of hospital acquired infection? Like wound infection, urinary tract infection, respiratory infections, bacteria and septicemia. So these are the main common types of hospital acquired infection. Now we will see about the management of hospital acute infection, how we can do the management. Identification, monitor for the signs. As a nurse, you need to monitor for the signs of infection, confirm with lab test and culture. So if you have the doubts, confirm with the lab test and culture. Antibiotic therapy, like prophylaxis antibiotics. So start targeted antibiotics based on the culture and sensitivity. So based on the culture report, the antibiotic should be started. Supportive care, provide fluids, oxygen and symptom management as needed. Infection control, isolate infected patients, follow strict hand hygiene and use of personal protective equipments. Surveillance and reporting, track cases and report to infection control teams. Also, preventive measures like sterile techniques, use the sterile techniques in hospital, proper catheter care and equipment disinfection. So these are the management where we can do generally the management of hospital acute infections. Yes, so next topic or next question, that will be 5S in safety protocol. So what are the main 5S here? First is sort. Sort, set in order, shine, standardize and sustain. So what are the main 5S? 5S's. First one, sort. What is sort? Remove unnecessary item from each area. So in a hospital area, remove the unnecessary items. Keep the necessary items only that is necessary, that you need to keep in a particular room or a particular ward or table. So that is sorting. So removing unnecessary items from the area. Next is set in order. Organize and identify storage for effective use. So for the effective use, identify and also arrange in a proper order. So arrange everything in proper order, that is set in order. Shine. Clean and inspect each area regularly. So that area should be shining. How? Keep it clean and infection free. Next is standardize. So this next fourth is standardize. Setting up procedures or performing these tasks. So setting up the procedure or performing these tasks, that is standardizing. Sustaining, perform this task on regular basis. So one day we've set the standard, we need to regular basis we need to use that standard, that is sustaining. So this is the 5 yes in safety protocol: sort, set in order, shine, standardize and also sustain. Next we will see radiation safety ALARA. What is ALARA stands for? ALARA stands for as slow as reasonably achievable. As slow as reasonably achievable. So what are the radiation safety principle? Time, distance and shielding. So what is time? Minimize the time you are in the radiation, like in the radiation room, like MRI scanning, like a scanning is CT scanning. So time, minimize the time near the radioactive object. Minimize your time. Next is distance. Keep your distance, maximum distance from the radiation area or radiation safety. For that, keep a distance. Shielding. So cover your body, protect yourself. If you need to go there, you can't able to avoid the radiation, then you need to use the shielding. Protective covering should be used. So this is the principles of radiation safety: time, distance and shielding. Minimize the time, maintain the distance and also use the shielding, protective covering or protective devices. Next we will see emergency code. So what are the emergency codes? Code blue is for cardiac arrest. So what is code blue stands for? Cardiac arrest. Code red for fire or smoke. So code red is standing for fire or smoke. Code orange, hazardous material or spill incident. So it stands for hazardous material or spill incidents, the code orange. Code yellow for disaster. Code white for evacuation and code green for emergency activation. So these are the main codes, emergency codes in the hospital. So code blue stands for cardiac arrest. Code red is for fire or smoke. Code orange for hazardous material or spill incident. Code yellow for disaster. Code white for evacuation and code green for emergency activation. Next important topic or question, that is biomedical waste management. So what is biomedical waste? It's a waste generated during diagnosis, treatment or immunization of human or animal. So the waste that is produced at the time of diagnosis, treatment or immunization of either the human or animal, that waste is known as biomedical waste. So that's the definition. So what are the sources of biomedical waste in a mainly? So the hospitals, clinics, laboratories, blood bank and veterinary hospitals. So from these are the sources of biomedical waste, like hospitals, clinics, laboratories, blood bank and veterinary hospitals. What are the types of biomedical waste here? So human anatomical waste, like body part and tissues. So human anatomic waste, like after the surgery, some body part and tissues. Infectious waste, cultures, swabs or bandages. Sharps, needles, blades and syringes. Pharmaceutical waste, like expired medicines. Chemical waste, disinfectants or solvents. General waste, like paper and plastic. So that is non-infectious waste. So these are the type of biomedical waste produced in a hospital or in a clinic or a veterinary hospital. So next, color coding of segregation. So how the waste will be color coded based on the segregation protocol in India in the year of 2016. So what are the color code here? Yellow. So the color yellow, in that yellow bucket, what the waste should be there? Human or animal waste or soiled waste. Like container is non-chlorinated bag, and how the treatment of this waste, human anatomical waste, will be treatmented? Incineration. So this is the treatment treatment procedure for the case of human anatomical waste. Next is red. In the red bucket or in red color, what are the waste should be there? Contaminated plastic like IV set or tubes will be there. It is autoclavable red bag and using autoclaving or shredding. So the treatment for this kind of waste is autoclaving or shredding. Next, white. White boxes will be there in hospitals too for sharps like needles and blades. It's a puncture proof box, should be puncture. White box should be puncture proof will be there. Autoclaving and shredding. So the treatment also is autoclaving and shredding. Next is blue. Glassware or broken bottles like medicine boxes and all, we will throw in the blue. Like cardboard box with blue liner. Disinfection and recycling. So the medicine boxes, after the medicine we draw out, so that will put in the blue. So this is the color coding of segregation in India based on the biomedical waste management rule in 2016. So color yellow is for human and anatomical waste or soiled waste, and the container should be non-chlorinated bag, and the treatment is incineration. Coming to the red, it is contaminated plastic like IV set, IV tubings. It is autoclavable red bag. So the treatment is autoclaving and shredding. White, it's for sharps like needles and blades. Puncture proof box should be there. Autoclaving and shredding. Blue will be glassware, broken bottles. Cardboard box with blue liner should be there, and a disinfection. It is mainly treatment is disinfection and recycling. Next, we will see needle stick injury protocol. So what is the needle stick injury protocol in a hospital? Immediate first aid. Wash wound with soap water. If you are getting a needle prick after using, that needle is used to a patient, it is very contaminated and infectious. You are getting a needle prick, what you need to do? So immediate first aid. Wash wound with soap and water. Avoid squeezing. Avoid squeezing your wound. Wash it with soap and water. Report. Should be informed to the supervisors immediately. Fill incident report. So you need to report it immediately. Next, risk assessment. Check the source of patient infection. So you need to check what infection the patient having, like HIV, HBC or HCV, any sexually transmitted disease or any human immunodeficiency virus disease that patient is having, you need to check that. Assess injury severity. So what injury you had, severity you should assess. Next is post-exposure prophylaxis. Start PEP if indicated. In the case of HIV, post-exposure prophylaxis should start. Hepatitis B vaccine or immunoglobulin if needed. So they need to take the vaccines if needed, like hepatitis B vaccine or immunoglobulins. Next, follow-up. Blood test for baseline and follow-ups. You need to go for the blood test. Your blood test should be done, and counseling and monitoring for the first 6 months. After the needle stick injury, 6 months, up to 6 months, you need to go for the counseling and monitoring. Documentation. Maintain record of incident and treatment. You need to maintain the record of incident and treatment about this needle stick injury. So this is the protocol for needle stick injury. So first, immediately first aid, you should wash your hands with soap and water. Then report. Risk assessment. Assess the risk of what infection the patient is having. Go to the post-exposure prophylaxis, follow-up and documentation. Next we'll see WHO hand hygiene steps with alcohol-based hand rub. So World Health Organization hand hygiene steps with alcohol-based hand rub. We are using alcohol-based hand rub. What is the WHO health hand hygiene measures? So apply a palm full of alcohol. So you should apply your palm full of alcohol based hand rub to cover all hand surface. Rub palms together. So after that you need to rub the palms together. Right palm over the left dorsum, back of hand. So use that with interlaced fingers and vice versa. Also rub palms together with fingers interlocked. So you need to rub back of fingers against opposing palms with fingers interlocked. Rub rotationally left thumb clasped in right palm and vice versa. Rub rotationally backward and forward with clasped fingers on right hand and left palm and vice versa. Also rub until the hand dry about 20 to 30 seconds. So if you are not hand washing, you are using hand sanitizers or hand alcohol based hand rub. This is the steps you need to follow. So what are the steps? Apply a palm full of alcohol based hand rub to cover all hand surfaces. Rub palms togetherly and rub right palm over left dorsum, back of the hand with interlaced fingers and vice versa. Rub palms together with fingers interlaced. Rub back of fingers against opposing palms with fingers interlocked. Rub rotationally the left thumb clasped in a right palm and vice versa. Rub rotationally backward and forward with clasped fingers of right hand in left palm also in vice versa, and rub until the hands dry about 20 to 30 seconds. Next is 3 key precautions while handling specimens. So what are the 3 key precautions we need to handle while handling? We need to think about while handling specimens. Wear personal protective equipments, PPEs, like gloves and lab coat to avoid contamination. First thing, wear the personal protective equipments. Label specimens properly. Next is label specimen properly with patient name and details, collection time to prevent mix-up. Avoid direct contact and spills by using leak-proof containers and carefully handling. So we should use leak-proof containers. So this is the 3 key precautions while handling specimens. Next is 5 elements of CLABSI. What is CLABSI here? Central line associated bloodstream infection bundle. So what is CLABSI bundle? Hand hygiene before catheter insertion or manipulation. So before catheter insertion, we need to have hand hygiene. Use full barrier precaution during central line insertion. So while you are putting the central line insertion, sterile gloves, gown, mask and drape should be done. Skin antiseptics with more than 0.5% of chlorohexidine preparation should be used. Optimal catheter site selection. Avoid femoral vein if possible. Daily review of line necessary with prompt removal of unnecessary catheter. So this is the 5 elements of CLABSI. What is CLABSI? Central line associated bloodstream infection bundle. Clear? Yes. So now we will move to the next question. Safety indications to prevent iatrogenic injury. So what are the safety indications? Verify patient, identify before any procedure or medication. So before the procedure, verify the patient. You are going to give the correct patient. Follow standard protocols and guidelines strictly. Use aseptic techniques to prevent infections. Double check medications and dosage before administration. Communicate clearly among healthcare team members. There should be proper communication between the team members. Monitor patient closely during and after the procedure. Document accurately all interventions and observations. Provide proper patient education about treatment and procedures. So this is the safety indications to prevent iatrogenic injury. So these are the measures. So next question, we will see about some MCQ questions, that is from both the microbiology and infection control. So the first question is the purpose of adding potassium permanganate to sitz bath is. So what are the purpose of adding potassium permanganate to sitz bath? So what is the answer? To enhance cleansing and provide antimicrobial effect. So it will help the enhancing cleansing and also providing antimicrobial effects. Next important MCQ, that is the common infection transmitted through contaminated medical equipment is. So what is the main common infection is transmitted from contaminated medical equipment to using to another patient. So it is methicillin resistant staphylococcus aureus, also MRSA. What is MRSA? Methicillin resistant staphylococcus aureus. So it's a common infection transmitted through contaminated medical equipment. So next question, the most common disinfectant used to clean pyogenic wounds. So what is the most common? It is hydrogen peroxide. So hydrogen peroxide is the most common disinfectant used to clean wounds, pyogenic wounds. The purpose of antimicrobial stewardship. So what is the purpose of antimicrobial stewardship? To effective treatment of infection. So it is to prevent effective treatment of infections. So next question, cholera vibrio was discovered by whom? So in the first itself, in the this class, we started with this question. So cholera vibrio was discovered by Robert Koch. Yes. So bacteria that die on exposure to oxygen are called what? So it is obligate anaerobes. So this bacteria will die to the exposure of oxygen. It is obligate anaerobes. Cutaneous infection is caused by Staphylococcus. So which cutaneous infection is caused by Staphylococcus? It is impetigo. Impetigo is a cutaneous infection caused by Staphylococcus. Next question, pulse polio immunization is a method of attaining. So what it is? It's a herd immunity. Pulse polio immunization is a method of attaining herd immunity. So these are some MCQs that is very important, and it's a previous year MCQs. So study well MCQ also. So this is very important. Yes. So we are coming to the end. We'll discuss about some exams, how you need to write the exam. So applied microbiology will be, so your applied microbiology and infection control and safety will be in 2 papers. So applied microbiology will be section A. So it is section A. So it carries us 37 marks, totally 37 marks this question. This is carrying, and MCQ. So a microbiology from MCQ will be 6. You will get 6 MCQ. So total 6 marks will come from the MCQ. And you will have a long essay. Long essay will be 1. So will be 10 mark. You will get 10 mark from 1 long essay, and short essay. So there will be 3 short essay, which will be 3 into 5. 1 will carry 3 marks. So it will be for 15 marks. So it will be, short essay will be 15 marks, and very short answers will be there. So for short answers, how many questions will be there? So there will be 3 questions which carry 2 marks. So 3 into 2, there is 6 marks you will get from the short answer. So this is the pattern of marking in the case, or question pattern in the case of applied microbiology section A. So totally 37. So MCQ will be 6, long essay will be 1 long essay will be there, and short essay will be 3, short answers will be 3. Coming to infection control and safety, it is section B. So that is carrying 38 marks. So total 75 marks. 37 for applied microbiology, and infection control is 38 marks. So here MCQs will be 7. So here MCQ. So 7 into 1. So total 7 marks will be you are getting into. 1 marks will be for each question. So 7 marks you will get from the MCQ. Also essay. Long essay will be 1. 1 long essay that carries 10 marks. Short essay will be 3. 3. So 3 into 5, 15 marks you will get from the short essay questions, and very short answer question. So short answers also 3 will be there, carrying 2 marks. It will be carrying 2 marks each. So it will be 6 marks you will get from the short answer. So this is the pattern of question division and marking in the case of applied microbiology and infection control. So total 75 marks will be, section A will be 37 and section B will be 38. Clear the marking and questioning? So any doubts till now? We have discussed about lot about this applied microbiology and infection control. Yes. So till now in the section we had discussed about applied microbiology and infection control, the most important and also important questions and topics. I hope everyone understood. So keep learning, keep growing with easiness where nursing education made easy. We will meet in the next sections with another interesting subject and interesting topics and questions. So till then, be with easiness, study well, write well the exams. All the best for your exams, and keep learning, keep growing with easiness where nursing education made easy. Thank you.